Welcome back to Cell and Molecular Biology course. I'm your course instructor Rini Tendral. In this video, we will see the differences between prokaryotic and eukaryotic cells. Before we learn the differences between these two types of cells, let's see the origin of life. Imagine a time before life, a time when Earth was a fairy ball. Yet within this chaos, the seeds of life were being sown. Simple molecules driven by forces began to interact, to combine, and to evolve. This is an incredible story of how life emerged from the primordial soup, a journey of billions of years that led to diverse and wondrous world we know today. The beginning of life and organic compounds formation. Our journey begins billions of years ago. A swirling cloud of dust and gas, remnants of a dying star, began to collapse under its own gravity. This swirling mass slowly condensed, heating up until it became a fiery inferno. This was the birth of our planet, a molten rock, slowly cooling and taking shape. Earth formed about 4.6 billion years ago from a cloud of gas and dust. It was initially a molten mass with volcanic activity and no oxygen. The early Earth's atmosphere was a far cry from what we breathe today. It was a toxic brew of gases, primarily composed of methane, ammonia, hydrogen, and water vapor. This reducing atmosphere, lacking oxygen, provided the raw materials for the incredible chemical reactions that would eventually lead to the first signs of life. This primordial Earth was a dynamic and energetic environment, frequently lightning strikes, powerful volcanic eruptions, and intense ultraviolet radiation from the sun. All these provided the energy needed to drive chemical reactions. These forces, though seemingly destructive, were crucial for the formation of complex molecules from the simpler gases in the atmosphere. This was proved by Miller-Urey experiment. In the 1950s, scientists Stanley Miller and Harold Urey conducted a groundbreaking experiment to investigate the origin of life on Earth. They created a closed system simulating the early Earth's atmosphere, a reducing environment rich in gases like methane, ammonia, and hydrogen. This mixture was subjected to continuous electrical discharges mimicking lightning. Remarkably, the experiment produced a variety of organic molecules, including amino acids, the building blocks of proteins. This landmark experiment provided strong evidence that the fundamental molecules of life could have arisen spontaneously from simple inorganic compounds under conditions thought to have existed on early Earth. RNA world hypothesis. A leading hypothesis suggests that RNA was the first self-replicating molecule, playing a crucial role in the origin of life. RNA, unlike DNA, can both store genetic information and act as a catalyst for chemical reactions. This dual function makes RNA a strong candidate for the first molecule of life. In this RNA world, RNA molecules would have self-replicated, evolved, and eventually gave rise to more complex life forms. Over time, RNA evolved and DNA emerged as a more stable and efficient molecule for storing genetic information. DNA, with its double-stranded structure, provided greater stability and accuracy in replicating genetic material. This transition to DNA marked a significant milestone in the evolution of life, allowing for the transmission of accurate genetic information across generations and paving the way for the incredible diversity of life we see today. Protocells. Over time, complex molecules began to form, including lipids. These molecules, when placed in water, naturally self-assemble into simple and spherical structures called protocells. These structures, resembling primitive cells, provided a crucial step in the evolution of life. They created a contained environment, separating internal chemical reactions from the external environment, allowing for the greater complexity and the emergence of early life. Cyanobacteria and the rise of oxygen. A major turning point occurred with the evolution of cyanobacteria. These remarkable microorganisms harness the power of photosynthesis using sunlight to convert water and carbon dioxide into energy. As a byproduct of this process, they released oxygen into the atmosphere. Initially, this oxygen was likely absorbed by rocks and dissolved in the oceans. However, over a millions of years, the relentless production of oxygen by cyanobacteria led to a significant increase in atmospheric oxygen levels, transforming the face of our planet. The Great Oxygenation Event. The rise of oxygen in Earth's atmosphere was a pivotal event. As cyanobacteria proliferated, oxygen levels gradually increased. Initially, this oxygen reacted with other elements such as iron in the oceans, causing massive iron ore deposits. However, eventually oxygen began to accumulate in the atmosphere. This great oxygenation event had profound consequences. It led to the formation of the ozone layer, which shields the Earth from harmful UV radiation, paving the way for evolution of complex life forms on land. The emergence of prokaryotes and eukaryotes, from simple to complex. Within these protocells, we saw earlier, complex chemical reactions began to occur. Molecules capable of self-replication, like RNA, emerged. These molecules, along with other cellular components, gradually became more organized and complex. Over immense periods of time, these protocells evolved into the first true cells, prokaryotes. These simple, single-celled organisms lacked a nucleus and other membrane-bound organelles. But they were the first living beings on Earth, capable of growth, reproduction, and metabolism. Endosymbiosis theory. A major turning point in the history of life was the emergence of eukaryotes. These cells, vastly more complex than prokaryotes, possess a true nucleus that contains DNA. They also possess membrane-bound organelles like mitochondria and chloroplast. The prevailing theory, known as endosymbiosis theory, suggests that these complex organelles originated from the symbiotic relationships between ancient prokaryotes. For example, mitochondria are believed to have evolved from bacteria that were engulfed by larger cells and eventually became indispensable components of the eukaryotic cells. The rise of DNA and cell complexity. DNA became the primary molecule for storing genetic information in both prokaryotes and eukaryotes, enabling more complex life forms to emerge. Let's see the differences between prokaryotes and eukaryotes. Prokaryotes are simple, single-celled organisms with no membrane-bound organelles and DNA not enclosed in a nucleus. However, eukaryotes are complex cells with a defined nucleus, DNA enclosed in a membrane, and eukaryotes also possess specialized organelles like mitochondria, endoplasmic reticulum, and other membrane-bound organelles. Evolutionary importance. The evolution of eukaryotes was a pivotal event, paving the way for the incredible diversity of life. The increased complexity of eukaryotic cells allowed for multicellularity, that is, eukaryotic cells could specialize and work together to form complex organisms like plants, animals, and fungi. They also allowed for increased cellular complexity. The presence of organelles like mitochondria and chloroplast significantly increased cellular efficiency, enabling more complex metabolic processes. They also enhanced genetic diversity. The evolution of sexual reproduction in eukaryotes led to the increased genetic variation, driving rapid evolutionary change. Without the emergence of eukaryotes, the intricate tapestry of life on Earth, with its vast array of organisms and ecosystems, would not exist. Let's see the overview of prokaryotic cells. What are prokaryotic cells? Prokaryotes are single-celled organisms with no true nucleus. Prokaryotes are smaller in size, that is, approximately 1 to 10 micrometer. The DNA is free-floating in the cytoplasm. They lack membrane-bound organelles. In the right, you can see the structure of a prokaryotic cell with the free-floating DNA molecules in the cytoplasm. And then you can see the ribosome subunits and the RNA and polysome. Now let's see the overview of eukaryotic cells. What are eukaryotic cells? Eukaryotic cells are those with a defined nucleus and membrane-bound organelles. The examples include animals, plants, fungi, and protists. The eukaryotic cells are larger in size, that is approximately 10 to 100 micrometer in size. The DNA is enclosed in a membrane-bound nucleus, and they also contain complex organelles like mitochondria. In the right, you can see a structure of a eukaryotic cell. The genetic material in this cell is found within the nucleus, which is surrounded by a nuclear envelope, And then you can see membrane-bound organelles like endoplasmic reticulum, which is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. And then you can see mitochondria and the Golgi apparatus, lysosome and peroxisome. So this is a basic structure of a eukaryotic cell. Now let's see the key differences between prokaryotic and eukaryotic cells. Let's start with the nucleus. Prokaryotic cells, like bacteria, lack a true nucleus. Their genetic material, that is DNA, floats freely within the cytoplasm. In contrast, eukaryotic cells, such as plants and animal cells, have a well-defined nucleus. This membrane-bound organelle contains the cell's DNA, keeping it protected and organized. Next, let's consider the cell size and complexity. Prokaryotes are generally smaller than eukaryotic cells. This smaller size allows them to have a higher surface area to volume ratio, facilitating efficient nutrient uptake and waste removal. Now let's talk about the organelles. Prokaryotes have no membrane-bound organelles. Eukaryotes, on the other hand, have numerous specialized organelles, including mitochondria, Golgi apparatus, and endoplasmic reticulum. As you can see in the figure below, you can see membrane-bound organelles in the eukaryotic cell, while the prokaryotic cell lacks the organelles. Now let's consider reproduction. Prokaryotic cells reproduce asexually through binary fission, while the eukaryotic cells reproduce sexually through meiosis or asexually through mitosis. Next, we will consider the ribosomes. Prokaryotes contain smaller ribosomes, that is 70S ribosomes, while eukaryotes contain larger ribosomes, that is 80S. As you can see in the figure, the largest subunit of the ribosome is 50S and the smallest subunit of ribosome is 30S in prokaryotic ribosome, which then assembles to form 70S ribosome. While in eukaryotic ribosome, you can see the largest subunit made of 60S and the smallest subunit made of 40S together assembled to form 80S ribosome. We will learn about ribosome in the upcoming videos of this course. Now let's consider the cell wall. Prokaryotic cells often have a rigid cell wall, typically composed of peptidoglycan, which provides structural support and protection. Eukaryotic cells exhibit more variation, such as plant cells have a cell wall made of cellulose, while fungal cells have a cell wall made of chitin. Animal cells, however, generally lacks a cell wall. Now let's see the flagella. Flagella are whip-like structures that extend from the surface of cells and enable them to move. The function primarily includes cell movement. They propel cells through liquid environment. In some cases, flagella can also act as sensory organelles, detecting changes in their environment. In prokaryotic flagella, the structure is simple, composed of the protein flagellin. They rotate like a propeller driven by a motor embedded in the cell membrane. While eukaryotic flagella are more complex, composed of microtubules arranged in a specific pattern, they move in a wave-like motion. Now let's see the examples. Prokaryotes include bacteria and archaebacteria, while eukaryotes include plants, animals, fungi, and protists. Now let's see the molecular level differences. About molecular level processes, we will see in detail in the upcoming videos of the course. Now let's consider the molecular level differences between prokaryotic and eukaryotic cells. Starting with the DNA structure, in prokaryotic cells, the DNA is a circular molecule often with extra small circular DNA molecules called plasmids. In eukaryotes, the DNA is a linear molecule packaged with proteins called histones to form chromosomes. Next, we will consider gene organization. In prokaryotes, genes are often organized into operons where multiple genes are transcribed together as a single mRNA molecule. While in eukaryotes, genes are typically transcribed individually. Coming to transcription and translation processes, in prokaryotes, transcription and translation occur simultaneously in the cytoplasm, while in eukaryotes, transcription occurs in the nucleus and the resulting mRNA is then transported to the cytoplasm for translation. Let's consider the introns. Introns are non-coding DNA sequences. In prokaryotes, introns are rare, while in eukaryotes, introns are common and are removed from the mRNA before translation. All of these are general differences, and there can be exceptions within each domain. Now let's summarize the points we learnt in this video. Nucleus. No true nucleus in prokaryotes and a defined nucleus in eukaryotes. When we consider size, prokaryotes are smaller while eukaryotes are larger and more complex. While considering the organelles, prokaryotes lack membrane-bound organelles, while eukaryotes have many of them. Considering the reproduction, prokaryotes reproduce through binary fission, while eukaryotes reproduce either through mitosis or meiosis. Their examples include bacteria and archaebacteria, while eukaryotic example includes plants, animals, fungi, and protists. Coming to the conclusion, these differences form the foundation for understanding life on Earth. Prokaryotes dominate in terms of numbers and survive in extreme environments, while eukaryotes gave rise to the complexity of multicellular organisms, including humans. Thank you. Hope this video was useful and you learned more about the origin of life and the differences between prokaryotic and eukaryotic cells. See you in the next video.